away, everyone. We're having some technical problems, but we'll get started now. And we're going to continue now with our discussion of working with whole genomes. And what I want to talk about today is picking up from where we left off last time on the topic of what we would do with a genome. So in particular, when we work with a genome, we're usually not going to be using what we get out of the analysis we talked about last time, so the raw genomic sequence. Usually what we're going to be working with is what's called an annotated version of the genome, where the annotations are basically extra information that's added to the genome that helps us understand what's significant and how we can work with that. So this is an example of an image from something called the Genome browser, in this case the NCBI MapView Genome browser, which is a way we would commonly access genomic information. We'll see more of that on Friday. But what I show here is some of these examples of tracks of annotation we would use when working with one of these genome browsers. Some of these are very straightforward, generic things. So this, shown from top to bottom here, is a track of annotation on genes annotated on this genome. So it's showing us where we believe there are particular genes and often adding various kinds of extra information on those genes. We also have another track here on RNA transcripts, a different transcript or splice forms that are known for these genes. Others may have more specialized uses. We have a track of repeats, so repetitive regions of the genome, different times annotated here, and some related to the process of mapping, so things that may be useful in refining this assembly or working within various kinds of specialized medical contexts. And some are quite specialized in their use. So this is a track of annotation that's particularly relevant to cancer studies. So this would be a track that would tell us about regions of the genome that tend to be gained or lost in cancers. And when we work with a genome, it's often not just the genome sequence, but these tracks of annotation we really care about. And on a modern genome browser, you may have dozens, even hundreds of these tracks of annotations you can choose from, as well as the ability to import your own if you need to make custom tracks for one reason or another. So what I want to do is talk a bit about the process that goes into getting some of these annotations on the genome. What I'm showing here is an image from the online text we're using, the NCBI handbook, that talks about some of the steps that go into getting a usable genome sequence online for their particular process. And this is a flowchart taken from the NCBI handbook that shows some of the major steps that go into getting a genome sequence onto the NCBI. And I'm not going to talk about every step in this, but I do want to illustrate a few of the basic steps that go into this analysis. One of the things that's very important to know is that usually there is some pre-processing that's needed before we even get to the genome assembly steps, because there are a lot of things that can go wrong, a lot of artifacts you need to deal with if you want even the basic assembly process to work. So very often you need to go through some steps that will screen out likely sources of error in the genome sequences. So very often you will end up with different kinds of contaminants hidden in your sequences that are going to mess up your assembly if you don't control for them. In the earlier technologies, we would often have to worry about contamination from the cloning vectors that we used. So you would get pieces of whatever sequences were in the cloning vectors that could end up in the genome. But even without that, we can end up with contamination from, let's say, a bacterium that happened to be infecting whoever you took your sample from, or maybe whoever was working with the samples, small viral genomes, various other kinds of artifacts of the molecular biology technologies. And you need to remove all of these things from your genome if you don't want to mistakenly assemble them into your genome. It's worth noting that this is not as straightforward as it seems, because sometimes things that look like this actually are in our genomes. There are many copies of remnants of viral genomes that are hidden in a human genome or any other complex genome we would look at. And sometimes in trying to screen out the actual contaminants, you may screen out something that actually is a part of our genome that looks like a contaminant. So you always need to be aware that errors are going to be an issue with your data. You can make your best effort to control for them, but it's very likely that you're not going to completely get rid of the errors. And you always have to be a bit skeptical when you look at these kinds of resources. We also often have to do work to screen out repetitive sequences. So for a lot of what we're doing in our analyses, we want to know where the repetitive sequences are. Sometimes we want to analyze them, but many of the basic analyses won't work correctly if we have common repeat sequences. So, for example, some of the techniques we depend on will be using sequence searching, BLAST, for example, and it's going to create problems with BLAST if we leave in common human repeat sequences when analyzing a human genome. Can anyone think of why that would be the case? Why are repetitive sequences a problem? Yeah.
If you blast against a part of that sequence, you'll get a ton of hits for every repeat, and that's not really constructive.
Yes, so you'll get a lot of hits that aren't meaningful. You'll get alignments to the repeats, which occur throughout our genomes and any closely related genomes. And a lot of the analysis we want to do has to do with aligning to other resources, maybe other genomes. So you need to hide these. You don't exactly remove them from your sequences, but you need to figure out where they are and put what's called a mask, which will screen them out from later analyses, even though you kind of leave them in your sequence. So all of that is kind of pre-processing and brings us to the actual assembly step. Some of this is kind of downstream from the things I was talking about in the last class. So we would have assembly of small pieces using overlaps of sequence, the shotgun assembly I talked about. But as I mentioned last time, we would usually have some incomplete assembly there, pieces that we can't connect to one another, and we need to do additional things to figure out how these are likely to go together. We may have other information from mapping, maybe from homologous genomes, other kinds of sequence we can throw into our analysis that will help us assemble things. And then all of this needs to be done to actually get to the complete assembly. But more or less, that would be the things we talked about last time, getting to a genome assembly. And that then is giving us a model of the base sequence of our genome, so what nucleotides are where. Usually there will still be missing pieces, misordered pieces, pieces you can't locate in the genome, but more or less that's getting you to the DNA sequence. And that's really the beginning of most of what I want to talk about today. So are there any questions about to this point of an assembled genome before we move on to the real annotation? Okay, so what we really care about at this point is figuring out what meaningful sequence there is in our genome. So what functions can we assign, or what other information can we correlate with our genome sequence? And there are a few basic methods for doing that. And actually, probably the most important one, and one that will get us very far in analyzing most of our genomes, is using alignment tools to do our annotation. So a lot of the annotation that comes into a newly sequenced genome gets there by aligning it to pieces of sequence that we've already annotated, or pieces of sequence that have a known structure or function to them. So in that diagram I was showing before, the flowchart of steps in the NCBI pipeline, we have a number of steps that refer specifically to this kind of sequence alignment annotation. So taking pieces of information such as RNA sequences we might have for our genome, ESTs, which are a specialized kind of RNA, basically short RNA sequences, models of genes, other known proteins that we might have come from our genome or a closely related one, and aligning all of that to our new genome sequence to try to figure out what's going on in that genome. For the most part, this is relatively straightforward. So if we have our newly assembled genome and we have a library of, let's say, mRNA sequences that we know come from that organism, and often it's much easier to get mRNA libraries from an organism than it is a complete genome, so we might have these sitting around. We can blast our RNA sequences against the genome and hopefully get some alignments that would look something like this, where we would have exonic regions, or excuse me, you would have the exons more or less lining up to pieces of the genome, and then you would have gaps where you would expect the introns to occur. We may have multiple splice forms for a given gene, so this might show two different splice forms that we observe in different RNA transcripts for this particular region. But those alignments, we would hope, would give us a very good model of what the likely splice forms are in this region of the genome. If you get a really high-quality alignment, probably what this is telling you is that there is a gene here, and that this is where the exons are found in different transcripts of this gene. So any questions about that? So it's worth noting that very likely that information is going to be incomplete. It may be that your library doesn't have every transcript of this gene you were interested in. You may not have a transcript library for the organism. It's not a very well-studied organism. And even if you have a transcript library, there's a pretty good chance that there are other transcripts you're not seeing. Maybe your library is based on some tissues and you have transcripts that only occur in other tissues. Or maybe it is based on particular points in time, and there might be other splice forms that occur at key stages in development or under key environmental stresses or disease conditions. So you never want to assume that what you see in your transcripts is everything that's present. So usually you use other kinds of methods to supplement that. Sometimes you'll have these EST sequences or other kinds of small pieces of RNA. So essentially non-assembled little pieces of RNA, typically raw reads off the sequencing machines, and you can form some models of where there are likely to be introns and exons based on where these align. So you might see that a lot of them align to some regions of this gene, and you could predict from that that those are probably what are called constitutively spliced exons. So exons that are always found in this particular gene in every splice form. And then you might have others that occur at lower levels that might be alternatively spliced exons. So sometimes they're there, sometimes they're not. You can't completely trust even this because it's not so unusual that you might not have any sequence aligning to some exons because you just weren't looking under the right conditions. And if you look long enough, you're going to see everything being transcribed at some rate. So eventually, if you get enough reads, you're going to get reads that are showing up in intronic regions, maybe just because the transcription or splicing process is a little imperfect. It sometimes makes mistakes in the cell, and it may be that there are reasons why something is rarely transcribed. So the information is going to be noisy, but it's still information you can use to come up with some hypothetical inferred models. So other splice forms you might propose might be there based on the alignment of these little pieces of sequence. When you get into actually working with these, it becomes very important to pay attention to the evidence behind your models. So we would often judge a model that has a sequence transcript supporting it as a high-quality model. There's direct experimental evidence for it. Others that we infer we might think are more speculative. Maybe they exist, maybe they don't. And if you were really going to work with them in a lab setting, you might want to do some experimental validation to see if they're really there. Is there any questions about any of this? So you can do more or less the same thing with other kinds of sequence. It may be that you have protein sequences you can align to your genome and see, based on these, do the proteins align in ways consistent with your gene models? You might be able to discover other gene models or other splice forms that way. And you can also take your gene models that you inferred from RNA evidence, and you can use those to predict protein sequence models. So if you think you know where the introns and exons are, that's going to imply what the protein coding regions are. And so that gives you a couple of different ways of getting annotated protein coding regions and annotated proteins. We can also use the techniques you learned last week about the domain prediction. So we can go straight into our genome sequence and try to predict conserved domains. And that would give us more information potentially on the functional significance of a particular gene, as well as more evidence for or against the existence of a particular gene in a particular region. But again, that's more or less using the same principles as we saw with aligning RNA sequences. Any questions at this point? All right, so one thing that you may remember I mentioned back towards the beginning of the class is that to a large degree today, protein sequences are things we get from prediction. Not too many people are doing Edmond degradation on the protein sequences anymore. So more and more often, the protein sequences are going to be coming from predictions from the gene models, which may be coming directly from the DNA sequence, or may be coming from aligning known RNAs to the DNA sequence. But that basically is taking us through how we can do genome annotation by alignment, and there are a lot of variations on this. So you can do annotation by alignment to other sequence you have on a given organism. You can align to another genome. If you're lucky, you may have another genome of the same species. More often, you might have a closely related species. And the more closely related your genomes are, the more likely it is the annotation will transfer. So you'll be able to say that the same genes with the same splice structure, more or less, will occur in the genomes, and aligning them to one another will give you good ideas of what's happening in your new genome. You can align to another organism's transcriptome, and the same kinds of principles will apply to a lot of other kinds of information. Not just genes, but also non-coding pieces of DNA, so what are known as RNA genes, such as things that code for transfer RNAs, ribosomal RNAs, microRNAs, and various other kinds of regulatory sequences. The same basic ideas will apply to all of these. So any questions about annotation by alignment before we move on from that? All right, so that is really going to be, for the most part, our primary tool for annotating a genome. You align it to things you already know about, and that can get you very far. It can get you very good annotations of a newly sequenced genome, and the more genomes we get and the more completely annotated they are, the better this is going to work. But it's not going to get us everything, and we therefore usually need to think about other methods that may fill in the missing pieces in our annotations by alignment. To tell us the things we can't infer by homology, and that's commonly referred to as Abinitio prediction. So predicting directly from the sequence without assuming we have any prior knowledge about homologous sequences. So there are a few reasons why we need to do this. One of these is that there may simply be no homologue for something we're interested in. It may be that we're looking at a gene that has never been sequenced before, or an organism that's so distant from other related ones that we can't detect the homology between them. It may be that we're looking at a gene that is found in other organisms but was never annotated in them. So we're almost certainly going to keep finding new genes even in a well-studied organism, and we may want a chance of finding these. We also want chances of finding things that may be wrong or incomplete. So sometimes our gene models in one organism are going to be incorrect, and we'd like a chance of discovering the correct ones, even if that's not known. And there's a very good chance that we're going to be missing information. Maybe there are splice forms we don't know about, genes we don't know about, and we want a chance of finding this. And for all of these reasons, we really want to be able to go into our sequences and find things that are not homologous to anything we yet know about, and that's kind of the motivation behind Abinitio discovery. So Abinitio can refer to a lot of different techniques. There are many variations on these, but I want to talk about a couple of techniques we would use specifically for the context of annotating genes. So as with many things in molecular biology, we would generally think about two versions of this. So gene annotation for prokaryotes and gene annotation for eukaryotes. And as is often the case, prokaryotes tend to be a lot simpler for these kinds of annotations. So in a prokaryote, we generally have a very gene-dense genome. They're generally under a lot of evolutionary pressure to lose anything that isn't functionally important, have a very compact genome that can be replicated quickly. And so prokaryotic genomes are almost all gene. And that tends to be a very useful thing for predicting genes, because it means if you're looking at a region of genome and you see something that might be a gene, there's a pretty good chance it's a gene. So that makes your prediction a lot easier. And they also don't have introns. So again, that's going to make things easier because your model of what a gene is going to be a lot simpler. So for prokaryotes, what's generally done is a pretty simple heuristic, and that is to look for open reading frames, or ORFs, and say that any ORF of a significant length, so let's say 200 nucleotides or more, is something we will annotate as a hypothetical gene at least. So does anyone remember what an ORF is? So an ORF refers to a start codon followed by a set of triplets, or a set of additional codons, and ending in a stop codon in frame. So basically an ORF would be something that looks like it might code a polymethionine and going to want to learn only stop codons. In some organisms, you won't necessarily have the canonical start or stop codons used reliably, but more or less this is the idea behind how prokaryotic gene prediction works. And when someone sequences a new prokaryotic organism, usually they'll just quickly run through and do exactly this analysis. Find all the ORFs and annotate every ORF of, let's say, more than 200 nucleotides as a potential or hypothetical gene. Many of those you can further annotate by homology to known genes, but you may end up with some of them where this is the only annotation. It's quite common when you look at prokaryotic genomes that you'll see lots of things labeled hypothetical gene or potential gene or something like that. Now it's worth noting that this is not always correct. We sometimes have non-canonical start codons. Those may be missed by a straightforward application of these techniques. And there's actually a lot of evidence now that the cutoffs people usually use, the minimum length cutoffs, are probably missing some important sequences. There may be quite a few more very short genes, essentially genes that code for little peptides, than was suspected even a few years ago. So probably these kinds of annotations are actually incomplete, and more of that is getting corrected and filled in as we learn more and study more about these organisms. But this gives you generally a pretty good first pass at a prokaryotic gene annotation. So any questions about any of this? All right, so eukaryotes are generally harder for gene prediction, and you should all, by this point, have a pretty good idea of some of the complexity that goes into a eukaryotic gene. So we've talked previously about some of the signals in the genome, so the signals that tell the organism that there is a gene in a given place, that let the transcriptional machinery and then later the translational machinery recognize that there is something to be transcribed and translated. But all of that contributes to eukaryotic gene prediction being a much harder problem in general than prokaryotic gene prediction. One of the big problems is that eukaryotic DNA is typically largely non-coding. A lot of genome is in introns and a lot of it is in intergenic regions. And that means that the intuition that things that look like genes are probably genes doesn't work when you're talking about eukaryotes. Just by random chance, if you have gigabases of DNA that isn't coding, you're going to find things in those gigabases of DNA that sort of look like genes but actually aren't. So you need much more specific models. Introns and exons mean that you can't use this intuition of just looking for long open reading frames. So you are generally not going to find long open reading frames even in real genes because you'll have the introns disrupting those open reading frames. So again, you need more complicated models. The signals that identify introns and exons are complicated, and we don't really fully understand how all of these work. Even an expert in transcription and splicing is not going to be able to reliably look at a DNA sequence and figure out whether that is a sequence of a gene or how that gene is spliced. So the cell doesn't always interpret these signals the same way, and we in our computer programs certainly can't always reliably interpret these correctly. We have other kinds of complications. So we have, for example, pseudogenes. So a pseudogene is something that looks like a gene but is not actually functional. Generally, the belief is that a pseudogene is something that was once a functional gene but has evolved to no longer be functional. Mutations turned it off, and in some way the organism was able to compensate, so it was better off with it disabled. Probably some of these actually are functional genes under certain circumstances, but usually we would think of them as kind of decoys, things that will distract our algorithms but not actually be genes. But what all of this is contributing to is that you need more sophisticated models. And that brings us into how you would do eukaryotic gene prediction. So most of the methods for eukaryotic gene prediction are based on a technique you saw last week, and that is hidden Markov models, or HMMs. And I want to pause for a moment because I think this illustrates one of the important concepts about computational biology I've been trying to convey over the course of this term, and that is that very often the same abstraction will be useful for many different problems. We saw this abstraction of hidden Markov models as a way of predicting functional domains of proteins. They're very useful there, but they're arguably even more important for gene prediction. The same kind of model comes up in this different kind of biological problem because in some sense it's solving an analogous computational problem. That's just an aside here. But basically what this is showing is a high-level picture of an HMM for detecting and interpreting gene data. So finding genes and annotating the intron-exon structure and various other features of the genes. This in particular comes from a paper that presented the first really successful eukaryotic gene predictor. This was called GenScan. And this is showing just kind of a little piece of what goes into GenScan. I'll show you a bit more shortly. This is showing in particular a high-level view of a model of genes detected on the forward strand. There's also kind of a mirror image version of this to detect genes that are being seen in reverse complement as you scan through a genome, because they can be running in either direction. And each of these little pieces here is actually hiding a more complicated HMM model. So each of these is kind of a box containing its own HMM to detect a piece of sequence that turns out to be important in annotating and detecting genes. So there's a box for detecting promoters, one for getting us started on 5' UTRs. We have a special path for handling single exon genes, a more common alternative path for multi-exon genes. We have this kind of complex structure here to handle movement back and forth between introns and exons. You need this kind of triplet model to take care of the complication of reading frame. So if you leave an exon in one reading frame, the next exon has to pick up in the same reading frame, and then various termination-related sequences to recognize that we've come to a 3' UTR and need a polyadenylation signal, and then can move back to intergenic DNA. So all of that is giving us kind of a high-level model of a gene. And the authors of this, Birch and Carlin, had to put together this model to recognize these key features of a gene so they would be able to identify in the sequence, does it have all of these parts that tell you that you're looking at a gene. And within each of these, we have even greater complexity. So, for example, I'm taking the box here that refers to the promoter, and that contains its own hidden Markov model, so a lower level hidden Markov model that describes what a eukaryotic promoter should look like. So there is, for example, a consensus model of a eukaryotic promoter where we would have a Tata box, so one of the important signals that would tell us we're looking at a eukaryotic promoter. A spacer, so this X with an arrow to it would represent an arbitrary length spacer. Another important regulatory sequence called capsite. And then if we could come up with models of those, that would suggest that by our sequence fitting these models, probably there is a good promoter of a gene here, or at least something that looks plausibly like a promoter. These would even have greater complexity, so the Tata box is hiding what's effectively a position-specific scoring matrix, basically a motif model for the nucleotide preferences for a Tata box and the surrounding nucleotides around it, and you need all of this complexity if you want this to work. You might notice that there is also an alternative path here. So genes would commonly use the consensus eukaryotic promoter, but we also have to allow for the possibility that we might be looking at a gene that doesn't have a consensus promoter. It has something we can't recognize because it doesn't quite fit the pattern for what a promoter should look like. Can anyone tell me what the point of this is? Why leave in the consensus promoter model if we're going to allow this to take this pathway where it just throws in an arbitrary spacer and skips it? What do we get out of having this model here? Well, all of these things come down to balancing different kinds of evidence that there is a gene here. We think most eukaryotic genes will have a consensus promoter, so if our sequence looks like it has one, that's good evidence that we have a eukaryotic gene, but some genes don't have the consensus, And if we allow that, that would generally be a low probability pathway. It's going to lower the likelihood of our overall estimate. But if we have really good evidence elsewhere in the sequence, so everything else looks really nicely like a gene, then the overall likelihood can be high. We can have a low likelihood in one piece because we've used the non-consensus model and make up for it with higher likelihood elsewhere in the gene. So all of this comes back to the fact that we need these models to often balance very complicated combinations of pieces of evidence, and we really need to automatically put together a model of the balance of the evidence, what that's suggesting. So often you need these kind of consensus models to usually get it right, but you also have to allow for the fact that many things don't follow the consensus pattern. So are there any questions at this point? All right, so there's more beyond this. This would be, for example, another one of these special modules, just quite a bit of complexity that goes into how we would code a single exonic sequence and capture all of the dependencies between nucleotides in different positions of a given codon, dependencies between codons, because all of this can give us evidence that would suggest that we're looking at a gene or not looking at a gene. But in the end, all of this is going to come together into a very complex model of what a eukaryotic gene looks like, with lots of these specialized modules that help us recognize key pieces of how a gene works. So all of these special pieces built into this model. Now, as you should have learned last week, we have algorithms for doing a lot of this automatically. We have techniques where if you give the model a good structure, basically the graph behind your hidden Markov model, and you give it example sequences, you can automatically train the model to recognize this, the sequences for that structure. So to figure out the probabilities of taking one pathway versus another, the probabilities of outputting particular nucleotides in different positions, so how often you see an A in the first site of a polydenylation signal and so forth. So all of that you can learn, but it's not going to work unless you start with a pretty good model, unless you start with a nice structure for your gene and are basically able to give the method lots of hints about what it should be looking for. And that illustrates another one of the key points about computational biology I want to get across here. So Burge and Carlin, in putting this together, could never have gotten this to work if they didn't know their computer science. So if they didn't know about hidden Markov models and know that there were techniques that could do this kind of analysis. But they would also never have gotten this to work if they didn't know their molecular biology really well. This really depended on having a very detailed model of what kinds of signals you were looking for, being able to give lots of hints to the algorithms based on your prior biological knowledge, and both of those things really go into successful computational biology methods. You've got to have a solid understanding of your biology and your computing to make methods like this work. That's another one of those general lessons I just mentioned as an aside. Any questions? Basically, we can end up with a model like this. That will give us a generic model for a gene. We can use that to train a model specific to a kind of organism. So we could train it to eukaryotes in general. We could train a model specifically for recognizing human genes or whatever we want. And then we have computer algorithms that will do most of the hard probability calculations for us. So that can take this model and a piece of sequence and can figure out the maximum likelihood way of threading the sequence through the model. So find the most plausible way of aligning the model to the HMM. And then we could say, if we're looking at a piece of sequence where every site aligns to intergenic region, we're probably not looking at something that has a gene. But if we're looking at a model where it's saying the most plausible, the maximum likelihood assignment involves threading through that whole gene model, then very likely we're looking at a gene. And by seeing which states of the HMM are assigned to particular nucleotides, we can get a detailed model of the structure of the gene and the regulatory sequences involved in it. So that basically is how we would use these. Now it's worth noting that these things are not entirely reliable. This is a very complicated kind of problem. These models work reasonably well and they keep getting better, but they're not perfect. So we might get about 95% of nucleotides annotated correctly with these kinds of genes. And 95% might sound pretty good, but when you start talking about whether you get even a single intron or exon right, you might get maybe 85% of your introns and exons exactly right. And if you're trying to predict proteins that come out of those introns and exons, getting it exactly right is pretty important. A small change can pretty radically change the sequence you think this codes for, so the protein that comes out of it. So 85% is not great. It's better than nothing, but this is not something people really trust. And very often what we would do when working with these is look at multiple kinds of annotation and compare these with other kinds of evidence. Maybe look at multiple predictions from different gene predictors, as well as looking at evidence from EST alignments, transcript alignments, and so forth to figure out what we really believe are the plausible models. These things are getting better, and very often where good ideas come from for improving these comes from a notion called comparative genomics, I'll talk about it shortly, which basically uses the idea that closely related organisms often have very similar sequences. So the first way this was really developed for these gene predictors was through a method called TwinScan. TwinScan was a method that instead of annotating one genome, would take two genomes simultaneously and try to annotate both of them at the same time. And the intuition behind that is that if these are closely related genomes, probably they have pretty much the same genes, very likely the same structure for those genes, and the annotation for one gives you evidence to support the annotation for the other. So if the annotations disagree, that's suggesting that maybe there is something incorrect there and one of them is wrong. If the annotations agree, you have more confidence that you're seeing a good annotation. And modern methods of this can be much more sophisticated, bringing in many different organisms and detailed models of how they're related, so phylogenetic trees on those organisms, to put together really strong predictions of what you believe and don't believe as far as the annotation of all of these. So these methods keep getting better, but they're not entirely reliable, and it's worth noting that that's especially true with respect to alternative splicing. These methods really don't work well at all for getting us rarer splice forms. They might get you the one best splice form. You really shouldn't expect that these are going to give you every splice form of the gene. So any questions about any of this? All right, so that brings me to the last thing I wanted to talk about today, and that is annotation by comparative genomics. And comparative genomics isn't really a specific technique. It's more of a high-level idea, this notion of using similarity between genomes to help us figure out what's significant or functional in those genomes. So we've seen some examples of this already in a sense. So we're getting comparative genomics when we do annotation by alignment. We're using the fact that we think information we know about one genome can transfer to another one. But there are much more special purpose tools that we can use to do very fine-scale annotation by comparative genomics. And this often comes into how the more advanced computational methods will work to try to get us better predictions by looking at multiple organisms at once. To illustrate this, what I'm showing here is a piece of genome from a genome browser. In this case, something called the UCSC genome browser. It's very popular for these kinds of studies. And what this is showing is running left to right, little piece of genome that happens to have here a piece of gene. So this blue thing here is showing us first an exon and then a little bit of intron for a gene, and the gene continues off to the right side of my right side of this screen here. And then what we have are some tracks of annotation related to conservation across different species in this region of genome. So at the bottom here, what we have is annotation related to conservation of this region of genome in a set of specific organisms. And there's a color code here that the black is showing us places where we have a very good alignment between the different organisms. Gray is showing us a slightly weaker alignment, and white is showing us where we don't seem to have any nice alignment at all. And what we have here is, at the top, this almost solid black bar is chimpanzee. So we can see the chimpanzee sequence almost perfectly aligns to the human in this region. We'd expect that because we think chimps are our closest living relatives. We have gorillas here. In gorillas, we have a nice alignment, and then this big piece where it's telling us there's no alignment. Can anyone think of why that might be? We would expect gorillas are actually the hemoglobin beta chain. That's a gene every vertebrate organism needs. So probably gorillas have a hemoglobin beta chain, and it's probably pretty similar to ours. So why don't we see an alignment here? Probably the reason for this is that the gorilla genome was in a relatively early stage of annotation at the time I made this image. Probably the gorilla does have a nicely aligning piece of sequence here, but there was a mistake or a missing piece in the genome. There's just a gap there that hadn't been assembled, and eventually someone would come along and fix that during the genome finishing process. But again, that's something to be aware of, especially when you're looking at new genomes. There are likely to be mistakes and missing pieces in them. And we can see that that's probably true because when we look at more distant organisms, we've got the next one is a monkey, a mouse, a dog, an elephant, possum, chicken. We still see very strong conservation. So almost certainly the gorilla is very close to us there. It's just missing information. The more distant we get, though, we can see the homology is breaking down. We still have versions of this piece of genome, even as far out here as the zebrafish at the bottom, but it gets more and more distant at the level of nucleotides. We also have this track of annotation here, which is a kind of summary of the overall conservation, a measure of how strongly evolutionarily conserved small regions of sequence are in the vicinity of this gene. And we can see that there's pretty good conservation within this exonic region. You kind of lose that shortly after the start of the intron. That's more or less what we would expect. The exonic regions are under stronger evolutionary conservation because it's important that this protein has a particular structure and a particular function that's implied by the amino acid sequence. Probably the intron here isn't really coding for anything important. It just happens that there's an intron there, and so we lose all of the evolutionary conservation. We can see that in more detail by taking a small region of this and blowing it up. So this is a region that's capturing a bit of the 5' UTR and some of the region above the gene or upstream of the gene before it starts. If I blow that up here, we can see a finer scale conservation at the level of individual nucleotides across these alignments. So what this green here is showing is at the level of individual bases, how strong does the conservation seem to be across all the mammalian genomes we had available for this analysis. And what you can see here are some interesting things. We see a peak of high conservation right around the start of the gene. So that, again, is something you would expect. We're getting into the coding region of the gene, or in this case, it's actually 5' UTR, but probably there's some important signals in the 5' UTR, and that's telling us here there's something important that's being conserved in that gene. We can also see a spike of conservation here, so this peak of high conservation that's actually before the start of the gene. So occurring about 25 to 30 bases upstream of the gene. Can anyone think of what that might mean? Why might we have conservation before the gene is starting? Yeah, so it's very likely that this is the promoter region of the gene. There are going to be important signals related to the transcriptional machinery recognizing that there's a gene here. In this case, based on where this is sitting and based on analysis of the sequence, we can say almost certainly what we're looking at here is a Tata box. So this is something we would expect to be about 25 to 30 nucleotides upstream of the start of the gene. And the sequence here looks a lot like a consensus Tata box. I know you guys can't see the sequence unless you're sitting right up front. But basically, we could say this is probably a Tata box. We could also look here, and we could say that there seems to be another peak of conservation here. This one's about 70 nucleotides upstream of the gene. This one we would again say probably there's an important regulatory signal here. Maybe less obvious what that signal is, but we can see that there's something that is strongly evolutionarily conserved at that site in the genome. And that kind of intuition is one of the key ways that people are discovering and annotating these regulatory sequences, which are often not nearly as well conserved and not nearly as easy to identify as, let's say, a gene itself. So we would look across our organisms, and by finding something that seems to be decaying at a slow rate evolutionarily, that's giving us a strong indication it has some kind of functional significance. Any questions about any of that? You can even do this kind of analysis at the scale of aligning entire genomes. So this is showing alignment of 2 different E. coli genomes. E. coli K12, which is a lab strain, O157H7, it's a pathogenic strain. And what I've shown here is what we get by running megablasts to align the entire eukaryotic genomes. And you should recognize these dot plots. We saw those back when we talked about blast. This is putting one little dot anywhere we've got a short piece of sequence with good alignment. And we can see here that we've got this diagonal from lower left to upper right here telling us that these are very close genomes. They're very similar. They align through most of the length of the genome. We do have this little piece here that's running diagonally in the opposite direction. Can anyone figure out what that might mean? Yeah, so it would probably mean that somewhere in the evolution of these, a piece of genome got flipped, what's called inverted. That's something that happens from time to time in evolution of a genome. So some big piece, a few hundred kilobases of genome, got flipped in the relative to one of these from the common ancestor. Here we can see a more distant alignment, E. coli K12 to Salmonella typhimurium. And here we can see that even though those are very different bacteria, they're actually pretty closely related. We see less homology at the level of individual nucleotides, but we can still more or less see that trace showing us that the bulk of the genomes is basically the same sequence and the same order, probably pretty much the same genes. We do have another one of those inversion regions, something more complicated. Then they represent a big piece of genome duplication, another thing we'll often see in genome evolution. But basically that's showing us how we can learn about high-level structure of a genome. You can even do this in large eukaryotic genomes, although you need more specialized tools. So this is, and there's a tool called Mummer, which would be an example of this, others that will work much faster than BLAST. So you can't really run BLAST to compare 2 eukaryotic genomes, like a human to a chimpanzee. That would be much too slow. But something like this will use the idea that you can very quickly find short exact matches. It's similar to the idea of the BLAST algorithm, that you can take your genomes and very quickly find pieces of genome that are exactly aligned, exactly the same nucleotides, and reasonably long, let's say 20 or 30 nucleotides. And by finding regions of genome that have lots of these exact matches, you can infer that there are large pieces of genome that may be conserved across organisms. And you can use this to put together models of how entire genomes may have evolved from common ancestors, this is called a computational karyotyping, where you can take a reference genome, color each chromosome, and then try to figure out, relative to that reference, how the chromosomes of any other genome are put together. This is showing some fairly distantly related mammalian genomes aligned to the human genome. And what you should be able to see is that even for fairly far apart organisms, basically across the entire mammalian portion of the evolutionary tree, you get huge regions of alignment, some megabases of sequence that at least at the level of genes and gene order are pretty well preserved across these organisms. So these are more advanced tools, but just so you're aware, these are things we can do and kinds of analyses that are available to us to get into these advanced models of how evolution works on very long time scales. So I know we're out of time here. Are there any last questions before we break for the day? Okay, so on Friday we'll get into actually working with some of the genomes and genome analysis.